Bio702 Physiological Systems of Animal. It is for MSc Biology 4th semester. The practical topic is determination of RBC's count. Significance of this practical in determination of the vitamin B6, B12 and folic acid deficiencies. RBC's count decreases due to deficiency of these vitamins, and due to internal bleeding and malnutrition it is again decreased. For high RBC's count, we can suspect smoking, heart disease, dehydration or hypoxia. Apparatus and chemicals that are required for this practical are hemocytometer, a microscope, lancet, cover slip and disinfectants. For chemicals, we require heme solution. Heme solution is composition of HCl, sodium sulfate, mercurial chloride and distilled water. And function of this heme solution is, because blood is thick, we have to dilute it first before counting RBCs, because RBCs are in millions. So this heme solution works as the dilution solution, and it also preserves RBCs and prevent clotting of the blood. And the apparatus and chemical required for this practical are lancet for pricking the finger in order to get blood, and then to get this blood in this micropipette. This micropipette is for RBCs counting, and this micropipette is divided into 100 equal parts. So we have to take about 0.5 parts blood and rest of the heme solution in order to dilute this blood, and the blood would be diluted for 200 times in order to count RBCs. We have cotton swab in order to disinfect finger, heme solution to dilute the blood, and we have hemocytometer, and it is Neubauer improved chamber for counting red blood cell, BCs and platelets and various other cells. Here are two independent chambers in the center of this slide. So we have to load this slide under microscope. We have to charge this chamber with the diluted blood and count RBCs under microscope. We can see the micropipette for RBCs counting. It has a red tubing, red graduation and a red grain in its bulb. This is to indicate that it is for RBCs. So what is the function of this bulb? This bulb is when we will take blood up to 5.5 parts and the heme solution up to 101 parts, then we'll shake the blood in this bulb, and this grain would act as a stirrer. It will help to homogenize the blood and heme solution. We can see here RBC's micropipette, it is having 1 equal parts of volume. So 0.5 parts will take blood, and then up to 101 parts will take heme solution, that is the dilution solution. Because we are now going to count RBCs in diluted blood, how much we have diluted this blood is determined through dilution factor. So dilution factor for this blood for RBCs is final volume divided by original volume. So the final volume is 101 and original volume of blood was 0.5. So the dilution is approximately 200 times. So we have diluted blood up to 200 times, and then we will count RBCs in this diluted blood, and then we'll convert it into concentrated blood by multiplying the counting with this dilution factor. We'll see it later. As we have already diluted blood, and now we have to put that blood in the Neubauer chamber. Neubauer chamber has two separate chambers in center, so we can do two countings at a time. We have to put a cover slip on one of the chamber and then load the diluted blood in it. Here we are looking at one chamber. So this chamber is actually 3 millimeter wide, 3 millimeter long, and it is a little depressed inside the slide. It has a depth that is 0.1 millimeter depth. So it can contain a volume of blood. How much volume it can contain? You can calculate its volume. Volume of the whole chamber is length into width into depth. So it is 3 into 3 into 0.1 millimeter. So volume is 0.9 millimeter cube. So in this whole chamber 0.9 millimeter cubes blood could be adjusted. So we have divided this major chamber into 9 equal squares. And now we have to calculate volume of one large square. So these are 9 squares. Volume of one large square is 0.1 millimeter cube. So volume of one large square is 0.1 millimeter cube. Then the central square is divided into 25 more small squares, and we have to count RBCs in these small squares that are in center. So in center there are 25 small squares. So this volume of one small square is 0.1 divided by 25. So it is about 0.004 millimeter cube. So we have to count RBCs in these 5 squares in order to conserve energy and time. So we have to load this chamber and count RBCs. How to identify this central square under microscope is that it is surrounded by all sides are linear lines, they are not squares. So we can identify this, and the most precise is we have to see 3-line boundary along this central square. These are 25 small squares, and we have to count RBCs in the corner square and a central small square. So if we count RBCs, for example, and a few of the RBCs, they may come on the boundaries of the squares. So how we decide these RBCs are part of this square or not? Either we should count or not. So for this we either count the RBCs on upper boundary and right boundary, and we will leave the RBCs on lower boundary and the left boundary. We'll use this rule for all the squares. Now we count RBCs in our experiment to see how many RBCs we can get in these 5 squares, and then we'll do calculation on those original number of RBCs. We have 0.5 parts of blood. Now we dilute this blood up to 101 parts. Wipe extra blood on the tip, and then we have to dilute it with heme solution. We have to fill heme solution. We have diluted it up to 101 parts, and now we shake it by rotating like this in order to make it homogenized solution. And now we'll wait for 10 minutes for the heme solution to work. Now we'll adjust this new bar chamber or monocytometer under microscope first, and then we'll load this chamber. Now we are going to put a cover slip on this, and then load the chamber. One of the chamber is covered with cover slip. Drain first few drops of the blood because it is diluted. Now we load this. By diffusion, diluted blood has gone between cover slip and the new bar chamber. Now we will count the number of RBCs. Here we have a counter for counting number of blood cells for differential and total count. So in first square we counted 90 cells, in second 104, in third 89, in fourth 72, and in fifth 119. So they make 474 in total RBCs in 5 square. As we saw previously, that volume of these 5 small square was 0.02 millimeter cube. Number of RBCs in 0.02 millimeter cube, they are 474 RBCs. So in 1 millimeter cube diluted blood contains 474 divided by 0.02, the volume of 5 small square. So it is 23,700 RBCs in 1 millimeter diluted blood. Now we have to calculate number of RBCs in concentrated blood. So we have to multiply this 23,700 with dilution factor. As we know, the dilution factor was 200 because we diluted blood for 200 times. 0.5 was the blood and 101 was the total volume. So we calculated this dilution factor, it is 200. So we have to multiply this 200 with 23,700. So it comes to 4.74 million RBCs in 1 millimeter cube or in 1 microliter of the concentrated blood. The normal ranges for RBCs count for men are 4.7 to 6.1 million cells per millimeter cube of the blood, and in women it is 4.2 to 5.4 million per millimeter cube. Our result shows that there were 4.74 million per millimeter cube of RBCs present, so they are within the normal range for men. So this is all about this practical.